Okay, so this part of the lecture will focus on process analytics with spectroscopic techniques. And with spectroscopy, you can see more fine details if you compare with the temperature measurement. So temperature measurements tells you that, okay, so now it's hot or cold, and now there's a change in the temperature, but you don't really know what is the chemistry behind of that change. So then it would be interesting to implement some of the infrared, near-infrared Raman techniques in the processing environment. So that would be interesting to know more about these processes. So you can measure with these tools, you can measure powders, solid material, you can measure liquids, but my examples, they are a bit biased towards solid examples. And I have here a few case studies where we are using near-infrared spectroscopy for looking into high shear granulation, and then also an examples of drying and then wet granulation of materials, and then interfacing with the Raman spectroscope. So this is the basic idea of doing process spectroscopy. So depending on the spectroscopic method, then you have a radiation source. So you need a light. So in case of near infrared, so this is a light that is having many wavelengths. But in case of Raman, your light source is a laser. So you have only one wavelength. And then that light you take to the sample by using fiber optics. So fiber optics are very narrow, so to say, optical fibers that can take light to the site of action easily. So if you are, for example, analyzing tablet, so then the light is interacting with your sample, and then you use again fiber optics to collect the light back to the spectrometer. So this is the cycle that is then done with process analytics. And when you are doing this, then you need to consider the optics here at the end of the fiber optic. So then you have a spectroscopic probe that is in contact with your material that is then illuminating the sample, and then you collect the light back to the spectrometer. I have a few examples of those later on. So this is an example that is quite often used for identification of material. We have these handheld instruments. So in this packet, we have everything in the same instrument. So this reasonable sized handheld instrument is containing the light source, and then there's some computer to analyze the spectral information. And then the light is coming out from here, from the top of the instrument, and then you control the measurement by using almost like this pistol type of solution. So then you measure the sample here, and then you have the data in your spectrometer. And this gives you already quite a lot of freedom to move around with this handheld instrument, and then collect properties of material. And this is commonly used for ID purposes. So here are NIR spectra of many different APIs. So we can see that there's a unique fingerprint of each of them. And then you can identify that you are working with the right material. And when you are interfacing with the processes, interfacing with the moving material, then there are a few terms that need to be defined. So you can do inline analysis, online, at-line, or offline analysis. And if we start from the bottom, so this offline is referring to a scenario, so to say classical scenario, that you collect a sample and then take it to the lab and then measure it maybe next week. And then you have your measurement. And then typical analysis is like maybe a wet chemistry like HPLC type of measurement. But these at-line, online, and inline, they are referring to scenarios where the instrument is closer to the process. So you bring the instrument to the processing environment, and then depending on a sampling solution, then you have different interfacing with your product. And this is, of course, important for the GMP purposes. So are you doing inline? Is your probe touching the material, or are you doing at-line analysis where you sample, but then your probe is not touching the product that will be then sold for the patient? This is all well defined in the pharmacopoeia. So you can look into the monograph 5.25 that is called Process Analytical Technology. And here you can see some of the terms that we have been already discussing here. So PAT is something that you do to achieve these timely measurements of critical quality attributes. And I would like to point out that here are also a few terms that are important. For example, real-time release testing is something that we would like to do for the product, that based on these process measurements, we would immediately release the product, and that would be ideal. And we also mention here continuous manufacturing. That is something that I hopefully have time to touch here also today. And here's a visual presentation from that pharmacopoeia chapter of these measurements. So for example, inline is visualized here where the probe is touching the process stream. And then this gives you a visual presentation of what these different terms mean. But please read this pharmacopoeia chapter to get more examples. Another related pharmacopoeia monograph that is interesting in the PAT context is this 2.9.47, demonstration of uniformity of dosage units using large sample sizes. So this is literally something where you have many samples, so more than 100. So this monograph here is presenting scenarios for having thousands of analytical results from a batch of your product. So you can keep on manufacturing tablets. So you, for example, have a batch size of 40 million tablets, and then you can measure a few million of those with a fast NIR measurement, and then you have a scenario how you should then release the batch based on this measurement. And this is interesting reading for real-time and real-life applications. But just to go to the spectrometers themselves, so this is a commonly used example of a process spectrometer. So we have here all the parts that I have been describing here for you. So here we have an example where we have a light source lamp is here, so you can see that it's there's some light coming here, and the sample is here on this distance. So you can measure the sample from the distance of 20 centimeters, and then the light is illuminating the sample, and then the collecting fiber is collecting the back reflected light to the spectrometer that is sitting here. And then this spectrometer is connected to a laptop, so then you can visually see the spectra after measurement. And this is another different type of probe. So this probe is having two different type of fibers here. So what we can see here is the end of the fiber. So this is the probe that contains then the few fibers that are bringing in the light, and then few fibers that are collecting the back reflected light. And then you can have this probe touching your material. So you can touch the tablet and then measure it, or you can touch the granules and then measure the granules. And that would be another way of interfacing the process. So of course these have to be made of stainless steel, so they are not getting rusty or any problems with when contact with pharmaceuticals. But these are something that can be used for measuring liquid, suspension, powder, many different type of samples. And now to these few examples. So the first example is the use for high shear granulation. And then this is a process that involves mixing granulation and then wet massing phase where we see the particle size. So in this first example, we are using near infrared installation. So what we have here is again the same lamp and something like a 0.5 kilo granulation equipment. So here we have 0.5 kilo of powder being mixed, and then we can add there a granulation liquid and then keep on measuring the process while it's going and without any sampling. So this light source light is here illuminating the granules, and then this probe here, this fiber here is collecting the back reflected light. And this we can use for understanding what is happening in the process. And then this is a result of a typical granulation. So what we can see is a mixing of powder in the beginning, followed by an increase in the water absorbance when we are increasing the aqueous granulation liquid. And then each measurement takes few milliseconds. So we can collect thousands of spectra while we are processing. And this is an area where you then start needing multivariate analytical tools. So you might have heard about chemometrics. So chemometrics is commonly used for analyzing these data sets. And what we can do is that we do techniques such as principal component analysis. So one spectrum becomes one spot here in a principal component presentation, and all of a sudden everything is easier to follow. So we can see the mixing of powder, spraying, aqueous liquid, so water addition, and then a particle size increase. So this is all possible to monitor with the NIR probe, and then also control in the later phase. And when you are interfacing with different processes, so then you start think about how the sample is looking like. So if you are fluidizing powder, it is having a different density than a powder that is flowing in a pipe, or a powder that is compacted into a tablet. And if you measure with the same tool, so then the optical density of your sample is changing. So then you interface with the same type of probe, so spectroscopic probe has the same light. So then the method is seeing a different volume, and this is known as an effective sampling volume. And you should always think about this, that how big part of your dosage unit you are measuring. And then there's a concept of this effective sampling volume, that it's increasing in this direction when the optical density is decreasing in this direction. And of course, your process spectroscopic tool is useless if you are only measuring the very surface of the tablet. So you have to think about what is relevant measurement for different cases. And then maximize the sampling volume so that you best describe something that is sized like a dosage unit. And then the concept scale of scrutiny is important here. So you need to have the scale of scrutiny close to the size of the dosage unit. And we can move on to seeing a bit more fascinating phenomena. So you can see also solid form changes during processing. So this is something that can happen for your material. So you have an anhydrous active pharmaceutical, and then you look it under microscope, it looks nice particles. But then when you take a droplet of water, all of a sudden crazy things start happening. So these compounds, of course, they have a tendency to dissolve the water, and then they can recrystallize. And if API has a possibility to exist as a hydrate, so then in aqueous conditions, it will transform into a hydrate with certain kinetics. And in this specific case, only in contact with 20 minutes, this compound was fully transformed. So you can see that it's starting to grow needle-like crystals. So this is, of course, annoying if you are during processing having aqueous components in the mixture. So if you are doing granulation, so then you add granulation liquid, and then this starts happening. And that's something that we have been investigating. And here is a bit more detailed analytics with one compound, nitrofurantoin. So this is the structure of the compound. And if you look on the crystal structure databases, so then you can find out that it can exist both as an hydrate and then as a monohydrate. So this anhydrate is having this planar packing, and then when it is recrystallized as a hydrate, then all of a sudden the packing motif is changing. So we have this characteristic herringbone packing of molecules. And this affects solubility, dissolution rate, particle appearance, and then ultimately bioavailability of the product. So you need to control this property during processing. And we can use Raman for doing this type of measurement. So here is an example of Raman spectroscopy for looking into different powder mixtures containing increasing amount of hydrate. So we can see that this hydrate is a good analytical signal. So we can see a change in the position of these peaks, and these are quite sharp features in contrast to the near infrared measurement. NIR spectral features, they are broad and quite difficult to interpret. But in case of Raman, we have sharper features that can be used for quantitative work. And that is indeed something that we did with this example, is that we picked up 4 different compounds that we knew that were forming a hydrate. So what could be done is that we do wet granulation for a few minutes, and in this case even up to an hour. And then we can see that these compounds, depending on their solubility and dissolution rate, they start transforming to hydrate. But there's quite dramatic difference in the kinetics of that one. So some compounds, they are slow transformers, and then some of them are fast transforming compounds. And this is relevant because typical wet granulation step, it can last, for example, 10 minutes, and then you can start drying here. And then it is a bit annoying if you end up somewhere in a halfway and then start drying from here, then you have very unstable product. And this is then important in the drying process. So this is an example where we have used very small fluid bed, only 2 grams of material here in this glass tube, but it's something where we can fluidize material. And since this one is made out of glass, so then it's easy to measure material properties during processing. And here we can get an idea how material is behaving. And in this specific case, we could look into the dehydration kinetics of this compound. So what we could see that depending on the temperature, then the dehydration kinetics were different. So this is something then what happens for your material. But this can even be more complicated, and that was the case with another model compound. So when we look into carbamazepine dihydrate that has this type of a structure where the water molecules are in the crystal lattice forming these tunnels. So then dehydration of this is quite dramatic because the structure will collapse. So you will have amorphous material for a while, which then starts recrystallizing. And in this specific case, we could see that carbamazepine dihydrate starts disappearing during fluidization. Then you can see the formation of amorphous matter, which then starts recrystallizing with the different kinetics into 2 different anhydrate polymorphic forms. So this is an extraordinary annoying case that your end product is a mixture of 3 solid forms. So this is something where you have to be very careful when you are wet granulating carbamazepine. And then this is a classical example with the effect on bioavailability. So many of these instruments, they are getting more smaller and smaller. So the concept of handheld instruments is something that is really like a handheld nowadays. So this is an NIR instrument that is the smallest on the market that I have managed to find. So it is really like a size of a matchbox, if you will. So with this one, we have everything in here. So the light, optics, everything is in this small packet, and you simply place your sample in contact with this part, and then you can measure NIR spectrum. And this can be connected to your mobile phone. Smartphones can handle spectroscopic signals, so that gives a totally new possibilities for doing process analytics. And another interesting application area is the falsified medicine or counterfeit medicine. So something that is either totally falsified product or something that is trying to mimic the original product. So with the visual appearance, you cannot see the difference between the original and the counterfeit product. They look the same, but something in the spectral fingerprint might be something that helps you in understanding what is the difference on this. So you can identify the falsified products by measuring with this type of instrument the NIR spectrum of it, or Raman spectrum. So this is indeed something that is important, and then there are many possibilities for looking into different processes, but all starts, of course, thinking about sampling. So how should you set up your measurement, and then how do you interface with the material? Are you doing inline, online, at-line, offline measurements? And there are many different tools out there, and then many different process spectroscopic tools, but they can, in ideal case, give you a totally new level of understanding of your system. So you can look into small fine changes while you are processing, and then really understand your process better. So my examples, they were related to wet granulation, but you could similarly look into like a protein expression during fermentation, or some separation processes, or synthetic reactions can be monitored with different tools into very fine details. But the basic principles are always the same. So think about the physical principle of the measurement, and then pick up the right tool that is intended for that purpose, and then consider this interfacing, and then do your measurements and then do proper data analysis. So that's a way to proceed. Good, but I think now we need a short break here. So let's close the lecture for a while.